So, next, bioresources refer to the living components of nature that can be used for various purposes such as food, medicine, and industrial products. Biodiversity, on the other hand, refers to the variety of life forms found in an ecosystem. The bioresource sector is a crucial aspect of sustainable development as it plays a significant role in maintaining the ecological balance and generating economic benefits. So, bioresources refer to the renewable resources derived from living organisms that can be utilized for economic, social, or cultural purposes. These resources include plants, animals, and microorganisms. The bioresource sector is essential for a country's sustainable development as it provides raw materials for industries, contributes to food security, and creates employment opportunities. The utilization of bioresources has been instrumental in the development of several industries such as pharmaceuticals, biotechnology, and agriculture and forestry. For example, many drugs are derived from natural products obtained from plants and microorganisms. Taxol, a drug used to treat cancer, is obtained from the Pacific yew tree, while penicillin, an antibiotic, is derived from the fungus Penicillium. Biotechnology companies use genetically modified organisms or GMOs to produce pharmaceuticals, vaccines, and industrial enzymes. The use of bioresources has also played a critical role in the development of agriculture as crops have been domesticated and improved over time to increase yields and resistance to pests and diseases. So, bioresources also play a vital role in maintaining ecological balance and ecosystem services. Plants absorb carbon dioxide from the atmosphere and produce oxygen, which is essential for the survival of all living organisms. Forests act as carbon sinks and help mitigate climate change by reducing the concentration of greenhouse gases in the atmosphere. In addition, bioresources are used in the restoration of degraded ecosystems such as wetlands and forests to improve biodiversity and ecological services. Despite the numerous benefits of bioresources, there are also challenges associated with their use. The overexploitation of bioresources can lead to the depletion of species and loss of biodiversity, which can have negative impacts on ecosystem services. The use of pesticides and fertilizers in agriculture can also have diverse effects on the environment and human health. Furthermore, the exploitation of bioresources can also result in the loss of traditional knowledge and cultural practices associated with their use. In conclusion, bioresources play a crucial role in the sustainable development of countries. Their utilization contributes to economic growth, job creation, and the provision of essential services such as food, medicine, and ecological services. However, the sustainable use of bioresources requires careful management to ensure the maintenance of ecological balance and biodiversity. It is essential to strike a balance between the exploitation of bioresources and for economic gains and the preservation of natural resources for future generations. Then, breeding in plants and animals. Breeding is the process of selecting and developing new and improved varieties of plants and animals with desirable traits. The goal of breeding is to enhance the genetic makeup of organisms to increase productivity, adaptability, resistance to disease, and overall quality. Marker assisted selection (MAS) is a modern breeding technique that uses molecular markers to identify and select individuals with desirable traits. Breeding in plants. Plant breeding involves the manipulation of genetic makeup of plants to develop new and improved varieties with desirable traits. The process of plant breeding includes the selection of parent plants, crossbreeding, and selection of progeny. The use of molecular markers has made plant breeding more efficient and precise. MAS helps in the identification of desirable traits, genetic diversity, and the elimination of undesirable traits. This technique is useful for developing varieties with traits like disease resistance, high yield, improved quality, and tolerance to environmental stress. So, examples of plant breeding using MAS include development of drought resistant or drought tolerant maize. Drought is a significant challenge for maize production. Breeders have used MAS to identify genes that regulate water use efficiency and drought tolerance in maize. These genes have been incorporated into maize varieties to develop drought tolerant maize. Development of disease resistant rice. Rice blast is a devastating disease that affects rice production. Breeders have used MAS to identify genes that confer resistance to rice blast. These genes have been incorporated into rice varieties to develop disease resistant rice. Advantages of MAS in plant breeding. Precision. MAS enables breeders to select individuals with desired traits precisely. Time saving. Mass helps to reduce the time required for breeding. Efficiency. Mass helps breeders to eliminate undesirable traits efficiently. Then disadvantages of mass in plant breeding. The cost of mass can be high, making it inaccessible to small scale breeders. Then breeding in animals. Animal breeding involves the selection of animals with desirable traits and mating them to produce offspring with improved traits. The process of animal breeding includes selection of parent animals. Crossbreeding and selection of progeny. Mass has been used in animal breeding to improve desirable traits like meat quality, milk production, growth rate, and disease resistance. Then examples of animal breeding using mass include development of disease-resistant chickens. Breeders have used mass to identify genes that confer resistance to diseases like Newcastle disease and avian influenza. These genes have been incorporated into chicken breeds to develop disease-resistant chickens. Then development of high milk yielding cows. Mass has been used to identify genes that regulate the blood production in cows. These genes have been incorporated into cow breeds to develop high milk yielding cows. Advantages of mass in animal breeding. Precision. Mass enables breeders to select individuals with desired traits precisely. Then time saving. Mass helps to reduce the time required for breeding. Efficiency. Mass helps breeders to eliminate undesirable traits efficiently. Disadvantages of mass in animal breeding. The cost of mass can be high, making it inaccessible to small scale breeders. In conclusion, breeding using traditional and modern techniques like mass is crucial for the development of new and improved varieties of plants and animals with desirable traits. The use of mass has made breeding more efficient, precise, and time saving. However, the high cost of mass remains a challenge, and particularly for small scale breeders. Bioremediation and phytoremediation. Bioremediation and phytoremediation are two types of environmental remediation techniques that use living organisms to clean up pollutants in soil, water, and air. Bioremediation involves the use of microorganisms such as bacteria, fungi, and algae to break down, to transform, or the harmful pollutants into less toxic or non-toxic substances. This technique is effective in cleaning up organic pollutants such as petroleum, hydrocarbons, solvents, and pesticides. One major advantage of bioremediation is that it is a natural process and does not involve the use of harsh chemicals that can have negative impacts on the environment. Additionally, bioremediation is often more cost effective than other cleanup methods. Examples of bioremediation include the use of bacteria to break down oil spills in oceans and the use of fungi to clean up contaminated soil. In the 1980s, a strain of bacteria called Pseudomonas putida has used to clean up a gasoline spill in California, which helped to demonstrate the potential for bioremediation as a practical cleanup solution. So, phytoremediation, on the other hand, uses plants to remove or neutralize pollutants from the environment. This technique is effective in cleaning up heavy metals such as lead, mercury, and cadmium, as well as organic pollutants. Plants can absorb and store these pollutants in their tissues or break them down into less harmful substances through a process known as phytotransformation. One major advantage of phytoremediation is that it can be useful to clean up contaminated areas without disrupting the ecosystem and can even provide additional benefits such as creating wildlife habitat or improving soil quality. However, phytoremediation can be a slow process and may not be effective for all types of pollutants. Examples of phytoremediation include the use of sunflowers to clean up soil contaminated with radioactive metals at the Chernobyl nuclear disaster sites, and the use of hybrid popular trees to clean up contaminated groundwater at a former industrial site in North Carolina. So, overall, bioremediation and phytoremediation are promising techniques for environmental cleanup that offer several advantages over traditional cleanup methods. However, their effectiveness can depend on a range of factors, including the type of pollutant, the environmental conditions, and the specific organisms or plants used in the cleanup process. Then, biosensors. Biosensors are analytical devices that convert a biological response into an electrical signal, which can then be detected and quantified. They have a wide range of applications in various fields, including medicine, agriculture, environmental monitoring, and food industry. In this essay, we will discuss biosensors in detail. So, types of biosensors. There are several types of biosensors, including enzymatic biosensors, immunosensors, DNA biosensors, microbial biosensors, and cell-based biosensors. Enzymatic biosensors use enzymes as biorecognition elements to detect analytes. Immunosensors use antibodies or antigens to detect analytes. And DNA biosensors use DNA as biorecognition elements to detect analytes. Then microbial biosensors use microorganisms as biorecognition elements to detect analytes. Cell-based biosensors use living cells as biorecognition elements to detect analytes. So glucose biosensors are one of the most commonly used biosensors in medical applications. They are glucose oxidase to detect glucose levels in blood samples. Another example is lactate biosensors, which detect lactate levels in the blood samples. Biosensors are also used in environmental monitoring to detect pollutants such as heavy metals and pesticides. In the food industry, biosensors are used to detect foodborne pathogens. Advantages of biosensors. Biosensors have several advantages over traditional analytical methods. They are highly sensitive, specific, and rapid. They can detect analytes in complex samples such as blood and urine. They are also easy to use and require minimal sample preparation. Then, disadvantages of biosensors. Despite their advantages, biosensors have some limitations. They require specific storage conditions and can be affected by environmental factors such as temperature and humidity. They can also be expensive and require skilled personnel to operate. Importance of biosensors. Biosensors have significant importance in various fields, including medicine, agriculture, environmental monitoring, and food industry. In medicine, biosensors are used for diagnostic purpose and monitoring of diseases. They have also been used in drug discovery and development. In agriculture, biosensors are used to detect pesticides and monitor crop growth. In environmental monitoring, biosensors are used to detect pollutants and monitor water quality. In the food industry, biosensors are used to detect foodborne pathogens and monitor food quantity and quality. In conclusion, biosensors are powerful analytical tools that have a wide range of applications in various fields. They have several advantages over traditional analytical methods, including high sensitivity and specificity. However, they also have some limitations, such as specific storage conditions and cost. Despite their limitations, biosensors have significant importance in medicine, agriculture, environmental monitoring, and food industry. Then, methods in biology, molecular biology, and recombinant DNA methods. So, molecular biology and recombinant DNA methods refer to the techniques used to isolate, purify, and manipulate DNA, RNA, and proteins to study their function and roles in biological systems. These methods have revolutionized the field of biology, leading to the development of various technologies that have numerous applications in medicine, agriculture, and industry. Some of the key techniques and methods used in molecular biology and recombinant DNA technology are discussed below. So, isolation and purification of RNA, DNA, genomic and plasmid, and proteins. The isolation and purification of RNA, DNA, and proteins are essential for studying their functions and interactions. Various methods are available for isolating and purifying nucleic acids and proteins from cells, tissues, or biological tissues or fluids. These methods include phenol chloroform extraction, column chromatography, and gel electrophoresis. So, different separation methods are used to separate and purify nucleic acids and proteins based on their size, charge, and other physical properties. Some of the commonly used separation methods are agarose gel electrophoresis, polyacrylamide gel electrophoresis, isoelectric focusing, and high performance liquid chromatography or HPLC. Analysis of RNA, DNA, and proteins by one or two dimensional gel electrophoresis, isoelectric focusing of gels. That topic. One-dimensional gel electrophoresis is used to separate and analyze proteins or nucleic acids based on their size, while two-dimensional gel electrophoresis is used to separate them based on both size and charge. So, isoelectric focusing is a technique used to separate proteins based on their isoelectric points. Molecular cloning of DNA or RNA fragments in bacterial and eukaryotic systems. Molecular cloning involves the insertion of a DNA or RNA fragment into a vector, which is then introduced into the host cell, such as a bacteria or eukaryotic cell. This technique is used to produce large amount of a specific protein or to create a recombinant organism. Recombinant DNA technology has numerous applications in medicine, agriculture, and industry, including the production of therapeutic proteins, genetically modified crops, and bioremediation. So these techniques have numerous applications in medicine, agriculture, industry, and other industries. That's all about this topic. Thank you for listening.